All right, next we have Livia Medney to give Genetics 101. Mrs. Livia Medney is a senior genetic counselor at Children's Hospital of Philadelphia and co-director of the Roberts Individualized Medical Genetics Center. She is a valued member of the Foundation Scientific Advisory Board. Today she'll be presenting Genetics 101, everything you ever want to know, but we're afraid to ask. I just added that last part. An introductory session designed to help us better understand the fundamentals of genetics and how they apply to ROR1. Please welcome Ms. Livia Medney.
Good morning. Good morning. Well, while the slides come up, thank you so much for the kind invitation. It is my pleasure and true honor to be here. And I think as many of us who were here the last couple of days, I come here for the fourth time now, and I'm humbled by all the experiences and the stories I hear. And we take them back, and it makes our work so much more meaningful. And I need to switch screens. Swap displays, yeah. And which one is this now? Can you come up here? Oh, yeah. So while we have it, I think for some of you, this is a review course, but I hear there are some new families, so please don't hesitate to ask any questions as we go along. Perfect, thank you. We'll just go over some of the principles to have all of us have the same kind of foundational knowledge about what we're talking about. I have no disclosures. And I think I'll take back, and this probably dates me, that the promise of genetics has been there for a few decades. And of course, this day and age, I should have had some TikToks up there, but I am a happily middle-aged person that still looks at printed material. But the promise has been there. And even though it feels that the therapy is not there, the progress has gone forward, and it is getting closer and closer for us to have a meaningful treatment. But it does all start, as you hear, about genetics. It will deliver on its promise, but it will take some time. And I think, unfortunately, and it's probably a reflection of the education people receive, even many, many physicians still have kind of this, oh my God, it's genetic reaction. But in reality, it's like any language or anything else you learn. Once you learn it, it's fairly straightforward, simple, and actually quite elegant. So I encourage you certainly to find a provider that will lower the bar to access clinical genetic testing and will also select the right tests for you. Implementation, especially in the US, of genetic testing is becoming more complex with our extremely complex system of healthcare coverage, which our European colleagues don't have to deal with. And there's increasing variety of tests that are an option for any family, and all of us who practice in this space have certainly seen test choices that were, let's say, suboptimal. So let's go back to genetics so we all learn the same fundamental basics of a language, right? As Dr. Banneman showed, there is really the basic central dogma of genetics. You have DNA, which is a blueprint that gets transcribed into the RNA, which is an intermediary sequence. And in the end, it gets translated in the protein. And for the case of RYR1, we have the gene, we have the RNA, and we have the ryanodine receptor protein. So the gene is a big gene. It's a 106 exon gene. So it's like having a book with 106 chapters. Think back when you read a 106 chapter book. It's a lot to get through. And you have the RNA, and it encodes for the ryanodine receptor protein, which is critical, as you heard and all know, for calcium release in the sarcoplasmic reticulum of the muscle. And to really simplify, my apologies to Bob and everyone who works, and Susan on the channel, right? So it is a gatekeeper of calcium channel release in the muscle. And it's so critical for the health of the muscle with then subsequent secondary kind of effects downstream. So back to genetics, what we're talking about. We have your DNA that's tightly coiled around histones into chromosomes, and they live in the center of the cell called nucleus. This is how we used to look at it. Luckily, this precedes me. This is like a circa 1960s. This is your entire genome at a way pre-digital resolution. And those are chromosomes, right? We all have the same chromosome, the same 46 chromosomes that generally have two X chromosomes in females, and most males would have an X and a Y chromosome, but others chromosomes are the same. You see the gray, white, black, what we call banding, and that is the same for all of us, kind of representation of the genomic architecture. And you get down at the DNA level. This is the core of the DNA language. I think everyone knows about the helix and that there is a ATGC, right? So adenine always pairs up with thymine, and guanine always pairs up with cytosine. And that's the beauty of this language, and also the fundamental ability then to kind of think about treatments that are small molecule derived therapies that take advantage of this. So basic gene structure. Genes are made up of introns and exons, and a gene has a promoter and a start and kind of the end signal at it as well. And again, I like to compare it to a book. Your exons are your chapters, the text that tells you the story. And the introns are kind of the illustrations in between. We don't fully understand exactly what they do. When I went to graduate school, the term of junk DNA was kind of around too, and like it's completely useless, and we know that's certainly not the case. We've all had the humble pie. So when the DNA is read, just like when you read a book, you can kind of skip over those illustrations or cut them out, and that process is called splicing, and the RNA, the intermediary molecule, then is just made up of the exonic sequences. That subsequently then get translated into a protein, which is, in this case, randine protein that's not quite working as well as we want to. And DNA also has its reading rules. As all know, there are 4 nucleotides, ATGC, but they're read in triplets. So if you do a very simple math, right, there's a possibility then to make 64 words or 64 amino acids. So 64 codons that encode 20 amino acids. If you think about it, there are other things like a piano. If you think, how many of you play piano, right? There are 8 notes in an octave, and a piano has 7 octaves plus 3 keys, right? So there's kind of many and infinite possibilities how to read the DNA code. And this is something that then is reflected when there are possibilities, and if you like to draw out and find yours or your child's DNA spelling error and what the changes are, this is a table that's readily available on any Google search, and I heard Dr. Google is a favorite here last night. So to bring it back, right, I said that it is as if the whole English language or any language was written in 3-letter words. So you have the word, the cat ate the mat, and the man got mad. All day his cat ate and ate, etc., etc. This is a Scottish writer who wrote this as a kind of little poem. So imagine if you change the C to a B, and all of a sudden there is bat ate the mat, and the man got mad. Well, this is a single letter or single nucleotide change that changes the word, or in the DNA languages, changes the amino acid, and we call it missense variant, right? Nothing is extra, nothing is missing, it's just changed. And some missense variants are tolerated. Our genome is full with variation. That's what makes us unique. So when they're benign or tolerated, the sentence still works, the protein still works. And whether the cat ate the mat or the bat ate the mat, the sentence still makes sense. And that's just like with some amino acid changes, they just are tolerated, they're fine. But all of a sudden, if I change C to an H and the hat ate the mat, well, this just doesn't make sense at all. And in that case, the amino acid change is not tolerated, or the missense change is pathogenic or likely pathogenic because the sentence no longer works. Now, distinguishing that is really extremely hard in many cases, but this is kind of the basic dogma we start out with. And then there are DNA changes that kind of disrupt how the sentence is read, right? We start a sentence with a capital letter. DNA reading starts with specific methionine codon, and it ends with a stop, or in language, a kind of period at the end, to take a breath and start a new sentence. So sometimes that stop or that period is in the middle of the sentence, and you can't read further. If you really are grammatically fanatic, you see a period, you stop in your thought. Right? So an early stop is something, again, it's often a single nucleotide change, and we call it a nonsense variant because nothing else afterwards makes sense, and that's certainly pathogenic. And then there are tiny deletions, all kinds of deletions of single nucleotides, 1 or 2 letters, and all of a sudden if we delete this C word, or the letter C, we shift the reading frame because we still have to follow the rules. It's read in 3s, so instead of cat ate the mat, it's the ATA, TED, HEM, ATA, etc., right? And we call this a frame shift deletion because something is really disrupting the triplet reading frame, and you lose that, and eventually it comes up to one of those 3 stop codons, and this is, again, a transcript that's generally subject to what we call RNA-mediated decay, so that RNA never gets translated into a protein. And they're all kind of, those are single nucleotide variations. There are other DNA alterations that we call copy number variations. They can affect the entire chromosome. If you have 3 chromosomes, here in this case, the example is 21, so that's trisomy 21 or Down syndrome. And one can also have small deletions or duplications within the chromosome structures that are not visible under the microscope. And we use kind of the microarrays. This is single nucleotide polymorphism or SNP microarray model, where we do take advantage of the more advanced technologies. And you can see the logarithmic ratio below 0. That's a deletion or loss of genetic material, and it can be as small as couple exons, couple building blocks, couple chapters from that RYR1 gene, or again, duplication of some of those exons. Both rare genetic mechanisms, but certainly we do see them as well. And then we have the DNA sequence changes and how they look. This is from Sanger sequencing, right? You sequence, you see it. This is, so to speak, what you hear, wild type, which is a reference sequence. And the lab always has to use a reference sequence, even though we know that our genome is full with normal variation. So those should be filtered out of any lab report. And you can have a sequence change, like here, instead of G, there is an A. And I think you can readily see that there is kind of this double peak in the readout. And it allows the lab scientists then to see that there is a change. Because of that. And then here's the frame shift deletion, it's the deletion of single nucleotide T, and you see single peaks all the way until the deletion happens and everything gets frame shifted. And then you see the double peak, and it's clearly evident that the variant is there. So to kind of quickly look at the nomenclature, what you see on your reports, the nomenclature can be written at chromosome level, which is not the case for majority of the individuals with RYR1-related diseases. The number is a number. Generally, there are 46 chromosomes. Then there's a sex chromosome complement, and any kind of differences in the other chromosomes are noted. So what we look at on most of your reports, you're looking at something that's describing the variation at the DNA level and at the protein level. So describing the sequence change at the coding DNA level of the gene is something that in your report starts with a C dot, and it has the nucleotide number. In this case, it's 8,801. Instead of G, there's an A. And then it typically follows by a description, what does that do for that protein, right? And it is describing the sequence change at the protein level of the gene. So missense change, right? P dot, instead of arginine, an amino acid position 2,934, there's a histidine. Then we have the nonsense changes or the stop codon changes that have various ways of being described on your reports, with an asterisk reflecting the premature stop codon in the most recent version of nomenclature guidance. And then there's sometimes silent changes where there's a change in nucleotide, but it doesn't change an amino acid. And those are particularly hard to interpret, even though you think, well, there's no amino acid change, there's more subtlety to the DNA because it can introduce the cryptic splice site as well. So this is kind of how to look at your reports, evaluate them, and think through what does it mean to have a DNA change, and then the harder question, how does it precisely affect the protein function? All right, and then we're going to quickly just go over some of the inheritance patterns of gene variants that in the prior, beforehand we used to call mutations. So there's dominant inheritance, right? It's a single pathogenic variant. And when you have that in one gene copy, it causes a medical condition and the individual is affected. And then there's recessive inheritance when one needs two pathogenic variants. They don't have to be the same, but affecting both copies of the gene. And then one is affected. And classically, which is not the case necessarily, as Dr. Bonneman alluded to in RYR1, a carrier is classically unaffected, which is not necessarily the case for RYR1. So if a parent has a dominant condition, there's a 50% recurrence risk with each pregnancy to pass on that gene change, but that doesn't predict automatically the degree to which someone will manifest any of the findings. We're not there yet. And if mutation is brand new in an individual and a parent is not a carrier, we still don't say never. We give about a 1% recurrence risk, and that's due to the possibility that there could be something called germline mosaicism. I just arbitrarily picked on sperm cells here, but they can also be in the egg cells, that there's a subset of egg or sperm cells that have this gene variation, and therefore this theoretical but real possibility of germline mosaicism is very rare, but certainly reported in pretty much all genetic conditions. So even before we knew about the RYR1 gene, the inheritance was recognized. It was known. This is a paper from 1975 of a large family from South Africa whose members have been here at prior conferences, kind of showing that it seems to run in the family, and almost half individuals get it and half don't. And as Dr. Bonneman showed, the muscle biopsies and ability to stain muscles and the ability to come up with the word central core was there long before the genetics came along, even though genetics now is kind of the primary tool. And it can go for years unrecognized. As I think, as you know, we both, Dr. Bonneman and I worked at CHOP, so we share a lot of the patients. And kind of when we saw this beautiful young girl who's been at conferences, who was, because of astute orthopedist, sent for evaluation because of hip dysplasia, and we recognized that it seems to run in the family. And even though her mom was diagnosed with cerebral palsy and a few other things, it was quite apparent that there is a shared inheritance, even though walking into the clinic, certainly I didn't know at the time. Dr. Bonneman knew that was most likely the central core disease. And the biopsy certainly supported that. Back in the day, the sequencing was much more difficult, but we were able to obtain it and showed that that is a 50-50 inheritance. But when we talk about, and I think some questions were asked, and the terms that you hear people use is something variable expressivity. It's when family members with the same genetic typo or variant will show different clinical manifestations. And in RYR1, there's also something called appearance of decreased penetrance. So some people who have the gene mutation or the gene variation pathogenic appear to show no clinical findings. But it, again, has to be taken with caution because of something called malignant hyperthermia susceptibility that you'll hear about more, which was classically thought of only as pharmacogenetic phenomenon, and now we know better, and we know that there are other triggers that can kind of lead to the similar reaction. So that's for dominant. And then there's recessive, right? If both parents are carriers of a recessive gene pathogenic variant, the recurrence risks are 25% that the child will inherit both working copies of the gene, 50% chance the child will be a carrier like parent, and a 25% chance the child will be affected and have 2 non-working copies. And that's the same for every pregnancy. It's not like if you kind of had one child affected, then only the other 3 options are for the next time. It's the same probability for each pregnancy. And for an individual who has recessive pathogenic variants, the recurrence risk really then depends on the partner's carrier status, right? If a partner is not a carrier, all the children will be obligate carriers. And depending on what those 2 alleles are in the recessive version, may show mild manifestations or no manifestations or have MHS risk. And if partner is a carrier, of maybe a truly recessive or silent variant, there would be a 50% chance for the recessive inheritance, and usually the more clinically significant involvement. So, as we alluded to, recessive is really complicated. Generally, it is earlier onset and more severe involvement. So here's a child who has a known missense pathogenic variant. And a novel variant, the frame shift, right? As we recall, mathematics, right, 13 is not divisible by 3, so therefore it cannot follow the triplet reading frame. It's novel, but we just know, and sometimes you don't need 20 patients with the same gene variation to know that this is affected how that DNA is read. It's a frame shift deletion, it's loss of function. And when we test the parents, we also show what's segregation because we can't always see that by sequencing. Although the newer technologies, the long-read genome sequencing, can kind of put the geography of the variants as well. So we know that mom carries the missense variant, which is known pathogenic variant for MH, and the father carries the frame shift variant, and it's causes, loss of function, or another term you hear as you, think about therapeutics, something called a null allele. And that shows that both copies of the gene have a variation, that it's truly recessive, and the segregation analysis is still quite often needed to show that. But then we have other recessive families, where again, there is a novel missense variant that we've reinterpreted as likely pathogenic, and a kind of del-C, so single nucleotide deletion, which is a frame shift deletion or a null allele. The novel missense variant was rightfully so interpreted as a variant of unknown significance by the lab, but we brought mom to clinic, and Dr. Bonneman found mild weakness and also had the typical muscle ultrasound finding, so clinically we can reclassify it then as being mild pseudo-dominant allele that can function also or show manifestations in a dominant or heterozygous state. And the other variant, it sometimes happens, but it's definitely possible, and that's why we recommend always testing both parents, is de novo. It's spontaneous. So that can happen, that both parents are not carriers. Not very common, but certainly can happen, and that was the case for this family. So typically, as I alluded to, carriers of recessive pathogenic variants are unaffected, but in RYR1, That's just not like many other things. It's just not going to follow that classic genetics, Mendelian genetics rules. Unaffected carriers can, not all, have a malignant hyperthermia susceptibility risk, and those are specific to missense variant carriers of missense variant, kind of the loss of function, null allele carriers are not thought to have the MH risk. And the apparently mild recessive can be a late onset dominant. So that's where it really, really blurs the concept of dominant recessive inheritance and the green and yellow peas we all learn about in high school biology, right? So in summary, pathogenic variants in RYR1 gene can act in dominant pattern, can act in recessive pattern, and they can cause a very broad clinical spectrum, right? There's variable expressivity, the broad clinical spectrum. There can be myopathy from congenital to adult onset. There can be various histotypes, as Dr. Bonneman showed, to various muscle biopsy findings. And we now certainly know there can also be triggered phenotypes, rhabdomyolysis, myalgia, malignant hyperthermia with various triggers. So how do you approach it? Andrew will talk about it. Sometimes genetics is very definitive and it's clear-cut and there's nothing to think through and it's definitive, and that when the genotype is interpreted according to the guidelines, and there's also often the segregation data that's needed. Hopefully you're seeing a neurologist who understands the phenotype and can really correlate all that. Sometimes we do need the muscle biopsy, which we call a histotype or histologic findings, kind of to support it. But as Dr. Bonneman said, everyone now starts with a genetic test instead of a muscle biopsy that's used as secondary supportive kind of criteria. And we often do absolutely need imaging, which is much less invasive than a biopsy. And it can be done by ultrasound or MRI. And we want and would love to have, and there are researchers here that are working on functional testing, which is currently only available for MH susceptibility, but that's on muscle tissue, which is quite invasive. And it was exciting to hear the talks about the functional assays being developed in research labs to assess for MH, and we hope that they would be there to assess for other variants of unknown significance, as well as other what we call omics approaches that are there currently in research settings. But all of those data points need to align to really arrive at a conclusive diagnosis. I think we all read the same literature. Dr. Marty's here, and we'll hear, this is a beautiful publication if you want to read, and I think it's on the RYR1 website as well, kind of talking about all the various future treatments, but they are guided by the genetic diagnosis. Some of them are very specific to the genetic variant. Some of them are, like the N-acetylcysteine that was there, right, could be more general to the muscle health and muscle well-being. But even you'll hear about the RICAL trial later on, that is also kind of genetic specific, because not every gene variant produces a leaky calcium channel. So that is extremely important that the genetics is defined, because that will guide future treatments. But while we wait for those, there are benefits to the diagnosis now, which is providing correct clinical management, correct clinical guidelines, and ending the diagnostic odyssey that those of you who are adults have lived with for decades maybe. It allows access to support, and we just heard truly heartwarming testimonials that finding the community that understands what you go through every day is just extremely meaningful. Those of us who see patients with the conditions, we can have knowledge, but we're not in your shoes. We can have empathy, but we don't fully understand what you go through. So the community support of this, I think, was evident yesterday or last night, and is certainly something extremely important. Access to research. We started this morning with research, and we'll continue to talk about research. Informing the family and kind of thinking about reproductive decision making, which is an extremely personal issue, and the answer to why. I think we heard some questions of why last night, and just having that answers really allows to work through that, even though that process certainly is not an easy one. So genetic diagnosis matters. It matters, and genetic diagnosis was what started this foundation when Carson and I saw Mike. As you can see, it's dated circa 2010. And because there was a genetic diagnosis, there is a foundation that now is looking back at 10 years of wonderful work and support and advocacy that is so needed. Like this day and age, no clinical trial, no effort can be done without the patient community. Those of us on the clinical or research side, we are informed by what you think, and every clinical trial should be informed by what the patient community wants and sees as important. Leaving patient community outside kind of those decisions is really ignorant. So the foundation did start from the genetic confirmation. So keep calm, do the genetic counseling, find a neuromuscular neurologist who understands what the spectrum is and can provide good guidance for you. And as you go through this, and as we come to these meetings, it's really humbling to recognize that you think you learned something, but by learning one thing, you're really raising 10 more questions. And we really, in this day and age specifically, should be open to discovery, because you really only are able to do it if you approach it with an open mind. I have the privilege of working at Children's Hospital of Philadelphia, where I've been for a quarter century now, and there's a wonderful genetics team, wonderful neuromuscular team, as well as neuromuscular team partners in pulmonology, and Dr. Myers is here today, cardiology, orthopedics, GI nutrition. We are privileged to continue the collaboration with NIH and Karsten's team, the foundation. Thank you for all the support you provide, and a shameless kind of plug. If you need to read more about it, we've put together kind of really brief 3 to 4 minute cartoons about genetics. That's the website, imgc.chap.edu, and it's really quick and easy, and if you need to send a family member there, please do so. Luckily, your taxpayer dollars have paid for even better visual aids, and this is through NIH. The website, maybe take it down, and there's beautiful educational materials about foundation of genetics. All right, thank you.
We have time for a couple of questions. Any questions for Livia?
We have a Q&A at the end.
Oh, we have a Q&A. Lindsay's reprimanding me, per usual. Okay. All right, I'm going to turn, quick question. Yeah, one quick question. Yep.
It's a quick question. My name's Stan Sollenberg. My son, 4 years old, has a R1 congenital myopathy. Both the talks this morning, so far the first two mentioned imaging and as a complement to the genetic testing, ultrasound and MRI. Just a quick patient history. We noticed some delayed milestones. We saw an orthopedist who diagnosed a hip dysplasia. And then at the suggestion of some family members that are physicians, we went to see a neurologist after that. They referred us to a neuromuscular specialist at your colleagues at Children's at Washington, D.C. Mm-hmm. They noticed hypotonia, but they were kind of pursuing two different paths, genetic testing and then also MRI imaging. They had suggested to do MRI imaging first with a child of that age would be, put under anesthetics. We had elected to go to the genetic testing first and then the MRI second.
Yeah.
But had we gone to an MRI with anesthesia, he would have, now we know, would have been at risk of the anesthesia with malignant hypothermia potentially. So just wondering if there's any clinical guidelines or, now in retrospect it's rather obvious, but to try to pursue the genetic testing first to try to figure that out if he would have been at risk of.
The genetics first approach, yeah.
Yeah.
I think a place like CNMC, I think would know that any child who is presenting for evaluation for possible underlying primary muscle condition would use safe anesthetics. Certainly all muscle biopsies in undiagnosed children are done with safe anesthetics with kind of removing those volatile gas anesthetics. So I think they probably would have followed, regardless, a safe sedation protocol. And sedation for anesthesia, and Sheila is here to, would have followed that. But genetics first approach is something certainly we endorse as well. And then ultrasound does not need sedation. The difference is that we lack neurologists who are trained in doing muscle ultrasound because it's not something that radiologists are trained to do in general. So these are specialty skills that some at CNMC certainly have. There's Dr. Barucha there who does muscle ultrasound at CNMC. So that ultrasound is the non-invasive way and really as an extension of physical examination in the hands of someone who's skilled to do it.
Pediatric anesthesiologist with an expertise in MH who might be able to opine on this. Is this thing on?
Testing. Yeah. One thing that scares the living daylights out of anesthesiologists in general is all you have to hear anything remotely close to malignant hyperthermia, and everybody's going to need Imodium for that. They're going to run for the hills. So administering a non-triggering anesthetic is simple, especially in an MRI unit. It is it's also the easiest way of administering anesthetic. So for those of you who are worried, all you have to say is risk of malignant hyperthermia, and immediately the smart ones will say, well, if we don't have the facility at this hospital, we will refer you to this hospital where they will administer a non-triggering anesthetic. I want to assure all of you that that's the standard of pediatric anesthesia. So please don't worry. All you have to say is malignant hyperthermia. Even if you're not sure, they're going to run for the hills and administer a safe anesthetic.
Thank you, Dr. Herlick. That's very helpful. Dr. Herlick is a pediatric anesthesiologist in Pittsburgh, Children's Hospital Pittsburgh, with an expertise in MH. He did mention Imodium. I have some if anyone needs any. We have a dispensary in the lobby. Question? Was there a question?
Hi, just two quick questions. Thank you for your presentation. It was wonderful. First question is, I was wondering if there's possibility that rare one disease is a multifactorial disease, like is only caused by the interaction of multiple genes. Like my wife has a diagnosis for rare one disease, but one of her brother has two genes that are mutated because he was tested by a genetic panel. One is GNS1. It is a similar gene that's in the pathway of rare one. But my wife only have a ROR1 gene mutated. So I was wondering if another heterogeneity factor can be caused by interaction between other genes. And the second quick question is if you think about the status of art, of science, do you think it will be possible in the future to understand the functional impact of a specific variant? If you understand the variant is on exon 5, for example, will we be able to understand the entity of the disease? These are my questions.
Great questions. Thank you for the first one. I think polygenic risk calculations is just an emerging field in genetics. I think it really depends what the gene does, and some are true recessives and true and unrelated, but I think we have to keep an open mind because polygenic risk calculations are a couple huge NIH studies looking at that for more common conditions. We'd have to look at it more on an individual level. So I think there's not going to be an easy answer, but obviously the human diversity and the beauty of it is explained by the polygenic kind of background. Your second question was about precisely predicting based on the gene variant location the outcome. Yeah, I think there are a couple talks coming up by Bob and Philip kind of to talk about modeling and doing that. Some of those variants, and that's the minority, can have pretty good predictions of outcome in terms of the broad spectrum, not narrow spectrum. But that's why there's going to be a database, and we need to continue to work together, and we're hoping with the RYR1 database project and the NIH-funded projects with ClinGen and ClinVar that we will be arriving at a more defined outcome prediction, which is never going to be absolutely specific to individual level because of that genetic background, right? This one gene is functioning, or this one gene variant is functioning on the background of 19,999 other genes or so. So I think the database project and those will give us better guidance, but we will never be precisely able to, I think, define the path year by year.
Thank you. Very insightful.
It was a great question because it has implications, obviously, on what treatments might work depending on the mutation and how it's affecting the RAD receptor. And so we've had conversations about that in the workshop the last 2 days. And so that fantastic question. I think we're going to wrap up. Let's thank Dr. or Mrs. Medney for the genetic counselor. Thank you so much. Wonderful talk.